My name's Neil Vickers. I'm at the University of Utah in the School of Biological Sciences, and I'm one of the on the leadership team for the PacVec Center of Excellence. And it's my pleasure to introduce two speakers today. And the first one is Samantha Sambado. She is a trainee, studying for a PhD in ecology, evolution, and marine biology at UC Santa Barbara, and she's under the mentorship of Dr. Sherry Briggs and Dr. Andy McDonald, and I'm going to let her tell you what she's been up to.
Perfect. Yeah, thank you, Dr. Richter. This is great. Hi, everyone. It's really great to be here in the PacVec community. Let's see if I can figure out my little screen. That should be fine. Yeah, so today I'm giving a talk that's titled Climate Associated Variation, Host Attached Juvenile Tick Phenology in California. There's like some loose themes around it, so let's just dive on in. So an outline for today's talk, a little preview. I'm going to give a big picture of tick-borne diseases, but the great thing about these groups is that people already know a lot about vectors, and so it's not not too many boring details. I am going to just intro the things I'm working on. A lot of these are manuscripts that are in various forms right now, so I'm just going to give you the very, very quick, brief overview of what they are. If you're interested in the methods, if you're interested in the results, please email me directly, and we can chit-chat about it as long as you like. I would like to acknowledge the PacVec collaborations. This is I've been part of PacVec since 2018, so it's been really great. It's been a year since I started my master's, and now I'm a fifth year of my PhD, so it's been a really special time. And then, yeah, there's a Q&A session. Maybe you can have questions, maybe I'll have answers, but yep, that's what the plan is. So big picture. Like many of you, I'm sure when you tell people what you do for your job, they're just like, okay, like what's my risk of XYZ disease? And then you'll probably do something like, oh, well, it depends, because vector-borne diseases, or this disease incidence that we're very interested in public health purposes, is really maintained by a variety of things, such as a host. Vector-borne diseases require a vector, it requires a pathogen, it requires something that can allow the vector and pathogen to connect over. And so when I think about how we think about vector-borne disease incidence, I think about all the different levels, and then more broadly in a macro scale, how things such as environmental drivers such as land use change and climate change, how that really impacts how we think about vector-borne disease incidence and how that gets reported. And so for today, I probably should have titled my talk what role does juvenile tick phenology play in each part? I'm going to talk about four many parts of this whole scale of things. Obviously, I've not figured out everything. Obviously, there's so many other facets within one layer. But for part one, I'm going to be talking more about the latitudinal gradients of tick abundances. So that's our vector abundance layer. Part two, I'll be talking more about the seasonality aspect of lizard-tick contact. So, your vector-host contact. Part three, kind of diving into more of, okay, if we have the ticks, where are they encountering hosts? And can we, map that out better with these data-driven recommendations? And then part four is kind of just throw it in there, why not? How does wildfire impact the tick system? And I kind of think about this in these parts 1 to 3 way. So part one, latitudinal gradient of tick abundances. So I think a lot about tick phenology, and because this is a vector-borne disease group, I'm going to say the ticks I'm referring to in this entire talk today are Ixodes specificus ticks. There's a lot of ticks out there, but that's what I'm focusing on today. And by phenology, I mean, tick behavior. How are ticks responding to environmental cues to then go out and seek for the blood meal host? And so tick phenology, I think about it as a within season dynamic. So within the couple months that ticks can be actively seeking out their host, thinking about the general patterns of their juvenile life stages. So juvenile life stages of ticks, I'm referring to the larva and nymph. And typically in California, we think of these two life stages interacting in a synchronous way, meaning in a given month, maybe the month of May, you see equally relative abundances of both life stages occurring at once. I do tend to say that tick phenology is not this binary outcome. It's not synchronous or asynchronous like it is on the East Coast, where there is a couple months in between the larvae and nymphs. There's a little more variation to even synchronous here in California. And so I thought about this in ways of saying, okay, where do we see these tick phenology patterns where either the nymph emerges before the larvae, or where the larvae emerges before the nymphs. And like many of you know, this order of when these life stages emerge can play a role in how these pathogens are maintained and then transmitted to the next life stage. And so it was really this Kurtenbach 2006 paper. I don't know who else has read it here. If I could see all your screens, I'd ask you to raise your hand. But it was really inspirational paper for me, where it talked about how tick phenology in the context of the East Coast, how tick phenology can drive differences in tick-borne pathogen risk, specifically Lyme disease risk, which is caused by the pathogen Borrelia burgdorferi, Sensu stricto. And so, you know, I see this hypothesis. There's many things that could be driving different tick-borne disease risks, but I see this hypothesis that tick phenology could be one of the driving mechanisms. And in California, tick phenology, like I said, is thought of as the synchronous phenology, where you have both life stages emerge at once. However, these juvenile ticks occur in a lot of different ranges of California. Like, California is a big state. There's a lot of different habitat types. And you'll find ticks, you know, from the Sierra foothills to Mendocino with all, you know, Bob Lane's group stuff. And even down here in like Southern California, beach ticks. They're kind of all over the place. And if we think that ticks are like us, they're ectotherms, they're very, you know, we're not ectotherms. We're very sensitive to changes in temperature and humidity. So if we're modifying our behavior by throwing on jackets or moving indoors or outdoors, ticks have to be responding in some way. And so instead of thinking about tick phenology as, you know, the same across our entire state that's very climatically different, I'm assuming that within these little regional pockets of California, tick behavior, tick phenology is probably more unique to its region versus the state as a whole. And I'm going to break up each of these parts into like motivation questions, methods, and the data. And we'll probably save questions for the end. So if you have a question, you can throw it in the chat, but I'll get to it at the very end. And so the questions, part one, I'm trying to figure out is, how much within season variation is there of tick abundances? I know I'm talking about phenology, but for this chapter, like this question is tick abundance specifically. And is this, you know, tick abundance variation explained by these latitudinal climate variables? So what I did to start answering this question is that on the right-hand side, we have a map of California. You can see each of these blue dots represents a UC natural reserve system location. So if you don't know about the UC NRS, it's really awesome. Highly recommend people go and check it out. But I was lucky to do a lot of my dissertation research out there, where I set up these 11 reserves ranging from Clear Lake all the way down to Malibu. And I would sample these 11 reserves in 12 days, once a month for 5 months, to try to capture as much climatic variation there is within the relatively similar time frame. And I set up these reserves kind of along this latitudinal temperature gradient. So north to south, you know, cold to hotter, and also this relative humidity gradient. So thinking about, if you're on the coast of California, you're experiencing higher averages of relative humidity, because I don't know, has anyone been to like Santa Cruz or the coast in June? You know, June gloom, it's a really thick marine layer versus the inland valley. Our inland, it's, you know, 100 degrees. So if we experience that much coastal difference in the summertime, I'm sure ticks are as well. And so what I'm expecting at each kind of region, whether it's like this coastal region or maybe a north to south region, I'm hypothesizing that it impacts the tick behavior. So you'll see these little phenology curves kind of a lot, where this x-axis is months. It's probably a scale of 1 to 4 months. And each curve represents the relative activity of an individual life stage, with the blue representing larvae and the yellow representing nymphs. And so we're hypothesizing in Southern California, where it's warmer, drier, ticks are responding to that. They're emerging earlier. They're having shorter activity, shorter seasons, versus ticks maybe in the cooler northern part of the state. They're emerging slightly later and maybe having longer seasons. So at each of these reserves, I set up 3 plots. At each plot, I would do 500 square meters of tick dragging. So taking long walks of the wood, looking at ticks off your cloth. And then I also would be lassoing about 15 lizards per reserve at each visit. So I kind of had these drag cloth ticks and these lizard ticks. And so I took all these ticks back to the lab, identifying the species. And like I said, for this talk, I'm only working on IPAC. I am testing these ticks for a different paper, but for this purpose of today's talk, I'm just talking about the tick count itself. With these field collected ticks, I then matched it to remotely sensed climate data. So the data it was selected, I pulled down PRISM data, and I pulled down things for like, you know, temperature, humidity, et cetera, threw all these kind of data into mixed effect models. So looking at the outcome is tick counts for a given plot as a response to these climatic variables with controlling for plot location. And so these are like really quick data takeaways. I'm not going to dive into all the data, but just to show that from this data, I could see that the juvenile tick phenology is not always the same everywhere. And what I mean by that is, so in these graphs, this x-axis is the months of March through June. On the y-axis is the individual 11 reserves, colored by brown for coastal, brown for inland, blue for coastal, and then separated by larvae and nymphs. And so if we assume that tick phenology was synchronous at the same time everywhere in the state, within these little red curves, you would think that all of the data would line up perfectly. You wouldn't have this much variation outside of this little red curve. So that's just to say, there's still some synchronous phenology, but it's not as intact, and it's not occurring at the same time in the same month at all these locations. The second thing I want to mention, people have heard me give this talk before, but your sampling method matters. So for me, I was really interested in the juvenile ticks. And so, historically in California, it's been very difficult to collect juvenile ticks with standard drag cloth methods. And so in this graph, I have, once again, months of the year, March through June. Y-axis is a log mean tick counts. The left-hand side is by lizard burden. The right-hand side is a sampling method by drag cloths. And we can see with lizards, if we're looking at the colors of life stages, that lizards is a great way to sample juvenile ticks. We actually only saw juvenile ticks on lizard, versus drag cloths are more primarily sampled the adults. So quick data, based on this empirical data of these 11 reserves sampled for about 4 to 5 months, phenology had a lot of variation, and it seemed like, how you sample kind of determines what life stage you can analyze. So that's kind of like the basis of like a lot of early empirical work I did. But now I'm going to hone in on specifically the seasonality of the juvenile ticks that were attached to lizards. So no longer looking at adults or drag cloth data, looking solely at the juvenile Ixodes pacificus ticks. And so I affectionately call this Project Pizza Party because these individual lizards, they feed a lot of these juvenile ticks. You can see here, this is a lizard that's in a little dental floss lasso, and these little gray gushers are, almost fully engorged ticks. And so in California, the juvenile ticks preferentially feed on lizards. And for this talk, I'm referring to the Western fence lizard, but they also preferentially feed on alligator lizards and things like that, particularly the nymphs. But the great thing about these Western fence lizards is that they're kind of found throughout California. So all of my sites from, Clear Lake to Malibu, there's tons of lizards everywhere, and they require less resources to sample. I could capture 15 lizards in a couple hours versus I try to do this with mammals. It's like a 3-day process, and you may not reach the same level of mean of tick burns as you do somewhere like on the East Coast. And so within this subset of data, I'm trying to ask, what is the relationship between these juvenile tick phenology patterns? So whether you have 2 life stages on one individual lizard, or is it all nymphs or all larvae, and how is that responding to some of these other climate variables? And so for this project, it's once again based on a lot of field surveys. So not just the field work I did in part 1, I also grouped up with some other people who had lizard tick data. And so here on this right-hand side, we have a map of California. Each point represents a unique location where lizards were sampled for ticks. And so in total, there's about 42 unique plots that were sampled at least more than 2 times in a given year. I then try to lump up this data into climate regions. So kind of looking at another way of looking at kind of breaking up California in an ecologically relevant way to what ticks may be responding to. So these climate regions are designated by the California Climate Change Fifth Assessment, where they have in these 5 regions. So we have like North Central California, the Central Coast of California, Southern California, and then San Joaquin in orange and Sacramento Delta in yellow. So from all these, I think there's about 1,500 individual lizards that were collected and about 9,000 individual juvenile ticks that were all larvae or nymphs. They were identified to at least, Ixodes species. And then similar to the first part, I brought down remotely sensed climate and land data from USDA and GridMet to then plug into these time series models. So time series models are now looking at kind of these tick phenology patterns, these tick counts as my outcome of interest, responding to these climatic variables. If we think there is some sort of serial correlation going on, these models may address or may pull out some of these climate variables that are important for our outcome of interest. And so some quick takeaways. So here in this graph, this data was also collected between 2011 and 2021. And so for each of those years, I pulled out the Julian date. So Julian date is the day of a year. So Julian date of 1 is January 1st. The y-axis is the mean ticks per individual lizard. And this is just, this black line represents like a GAM model showing that the mean ticks a lizard as a whole for nymphs is around, Julian date 112, which is kind of early or mid-April. And then for larvae, it's kind of around 1/10, a little bit of a bump around 135. And so if you look at when the maximum mean tick per lizard occurs, so at a given climate region, these vertical lines represent climate regions, and they represent when the max abundance occurred at that given region. We can see that this red line here, this is southern coast, so they reach their max pretty early. Day 85 is usually late March, versus here, like around day 112 is about late April. So we can see that these southern nymphs are reaching their max levels earlier in the season. And I do think this is like an interesting data point. I would love if someone was able to test these nymphs. Like, I'm not quite sure if these are unsuccessful nymphs from the prior season, or if these are newly emerging nymphs. So I think that's like the next level that someone should investigate. But with my data, I'm still seeing that nymphs are emerging before larvae in some climatic regions. And through my time series models, I'm seeing that climate regions with higher mean max temperature tend to see an increase of juvenile synchrony, so both larvae and nymphs feeding on the same time on an individual lizard, which supports some of Andy's earlier work. Okay, how are we doing on time? I did talk to Kacie. So Kacie and I decided to split this up differently, where I'm going to give a slightly longer talk and she's going to kind of briefly introduce her talk, but we're going to get through my talk eventually. So for part 3, this is kind of what I've been newly funded on for PacVec. This is kind of looking at quantifying human contact with ticks. How can we use data-driven strategies to reduce some of these contacts that are happening in California? Oops. So motivation question. So a lot of, California tick ecology and theory, despite just like, the mega studies that Bob Lane's group has conducted in Mendocino, there's been a lot of tick theory that's been like borrowed from the East Coast, because on the East Coast, that tends to be higher prevalences of Lyme disease and greater tick-borne disease risk. But however, we know California, the tick system is very different in terms of like climate and vectors and different pathogens that are present. And so this proposal is trying to say, okay, what are strategies to reduce tick contacts here in California, which we know is slightly different than Northeast. In the Northeast, people tend to encounter ticks in their backyard while they're gardening or like crossing their front lawn. But here in California, there's been some literature to suggest that people are encountering ticks not as much in their house, because we don't own McMansions. They're encountering them more when they're out in public land. So when they're hiking in Sonoma or Point Reyes. And so there's more of this like public land interface that people in California experience in ticks. And so, but really, like, there's no tick-borne disease without humans. Like, yes, we can know where this is tick suitable habitat, but if people are not visiting it, then that probably shouldn't be where we focus a lot of, vector control efforts to reduce these potential tick contacts. And so building on this empirical data from my previous research, we're now also trying to overlap where human activity is. So, humans are very prone to overshare their geolocation. There's a lot of publicly available data, whether that's Strava, Twitter, Flickr, cell phone data. You can then start to use some of this human-generated data to overlap with this tick empirical data to try to have a better idea of where humans are recreating and if they're recreating over an potentially infectious tick area. And so, like I said, the question here for this project is, what is a seasonal geographic overlap between tick suitability land and human prone areas where they're more likely to recreate? And can we use this data from the tick side and from the human side to create some strategies to reduce human tick encounters? And so this is a proof of concept. This is just a risk map. And so this is from Andy's 2019 paper that other people on this call have worked on. The idea is to create some sort of risk maps like this. This map was actually only for adult ticks, but I like to do it for the juvenile ticks, particularly for the nymphal ticks, understanding their seasonality. So bringing in all the adult and juvenile ticks to build these species distribution models with a human layer component to it, where we're scraping this human geolocation data And then we can try to build these risk maps that are based on a month to month and also more county to county instead of just all the state. And so hopefully from these risk maps, we can then try to collaborate and build in some public education programs a lot of people are already doing in the state, but just say, hey, like there's this trail in the Bay Area. We know there's infected ticks. We know tons of people use it on the weekends and weekdays. You know, maybe we should try to mow that area more frequently than maybe mowing like Pinnacles Park where, you know, there may be ticks there, but no one really visits it. So we shouldn't put a lot of effort into maintaining that as much if we have a limited number of resources. And so some expected outcomes of this project is to develop this novel method to quantify human tick contacts through this, you know, scraping of human geolocation data to kind of build some of these affordable environmental interventions. So such as like signage or informing, you know, park rangers, hey, this is what's going on. Trying to highlight, to target and optimize some of these tick suppression applications that can be used. And once again, you know, the purpose of PacVec is to kind of strengthen these linkages and collaborative relationships from, you know, the academic to outdoor enthusiasts, which also a lot of academics are outdoor enthusiasts as well. And lastly, this will be real quick. So, okay, we have an idea of like, there's a lot of different, you know, tick system is not one monolith in California. There's a lot of pockets of unique nuance to either just general abundances or how the juvenile life stages are interacting with each other. We also know that humans interact with the landscape differently. You know, it was concentrated more around urban centers versus more the rural part of the state. But another thing that we have to keep in mind is that we live in California. If you're a California ecologist, you have to think about fire because fire it's here to stay. It's always been here to stay. And so kind of thinking about wildfire as it's a significant part of California ecology, and wildfire tends to also occur where ticks are, meaning a lot of wildfire occurs in, you know, oak woodlands and, you know, grassland habitats. But not all fires are the same. And what I mean by that is that a wildfire can vary in size and also heat intensity. And so when I was out sampling ticks in 2021, I was slated to go to all, you know, 11 of these reserves in January, but 4 of them actually caught on fire that fall of 2020. So it was a very just kind of like come to Jesus moment. I was like, okay, well, you know, I might as well also sample this because fire is part of this landscape, and I'm sure it's having some sort of impact on the ecosystems that support the ticks that we're trying to figure out these long-term trends to. And so for this project, I'm thinking about what are the direct and indirect effects of wildfire on tick populations. And so for this project, these are separate field surveys, so not field surveys that were associated with my parts 1 to 3. I went out and collected a lot of tick drags just at these 4 reserves. Each reserve had 30 plots, 15 burned, 15 unburned. And within those subsections of 15, there was 5 replicates of 3 vegetation types. So there'd be 5 grasslands, 5 woodlands, 5 shrublands within the burned and unburned plots at these 4 unique reserves. So 2 in Northern California and 2 in Central Coast California. I got really lucky where while I was out there, I also connected with the UC Santa Cruz team led by Gage Deaton and Kelly Zilkis, where they were going out and sampling basically everything post-fire. So they were going out and sampling like the carbon in the soil, the height of vegetation, like how charred was the soil, how many inverts were left over. So they did a lot of this bulk of like actual empirical like environmental sampling of all these fires, and they did it for 3 times. So right after the fire, 6 months after, and then 6 months after that, which overlapped the time when I went out and sampled these fire sites 6 times in a matter of 2 years following those fires of 2020. And so once again, I have this empirical data that I collect on the tick side. This UC Santa Cruz team has shared with me all of their fire empirical data. And then I've been pulling in a lot of remotely sensed climate land fire data to build up these structural equation models, which is just trying to understand these like kind of causal effects of more than just, okay, we have burned and unburned ticks, which are probably both influenced by, you know, the climate of the given region, but we also have to consideration the fire severity for some of those ticks. And maybe there's other, you know, unique characteristics that are shaping the abundances of these given reserves. And so, I don't know, the firework is kind of, for me, it's like, we tend to see things that make a lot of sense, like, you know, plots that were, so this is a graph where the x-axis is individual reserves, so the 4 reserves that were sampled, y-axis is the log tick counts, And then we have it separated into the burn treatments and unburned treatments. And so, treatments that were not burned, we tend to still find we still tend to find ticks, which is why we don't see as many, low 1 to 2 tick counts. There tends to be some impact of like, okay, sites with higher percent of char soil, we tend to see lower percentage of quadrants for vegetation, we tend to see lower tick count, which also makes sense. And then, the grass, although the grass burned, it replanted and regrew like normal the next year. So those areas, even though they were burned the prior year, they tend to act like normal grass plots, versus places that were like oak woodlands that burned. It took about 2-3 years for that tick population to actually return. So there's a lot of things going on, but a lot of like very normal outcomes that we would expect for some of these fire plots. So that was a lot of different projects in like 30 minutes. But yeah, that was kind of just a flavor of here are these things that I'm working on. Like I said, they're in various forms of manuscripts. So if you have more questions about any of the methods and data, please just email me separately and we can just, talk more about it. But I did want to mention some other projects. Like I said, this is my 3rd time being funded by PacVec. I'm really appreciative of this community. And like what I love about PacVec is that it can be very collaborative. And some of these collaborations have resulted from other publications from other people that are part of Sway Lab. So if you're interested in maybe more of the the rodent parasite community stuff, that was like a lot of work I did in the Sway Lab. That was recently a new paper in Journal of Parasitology. Last year, Andy, who's now a new member of PacVec, he led a paper about linking the Lyme disease ecology and epidemiology. So, where are the infected ticks? And if we can find infected tick suitability, should we also see higher rates of Lyme disease as well? And then the paper that I think most people on this call were on was led by Chris Paddock about isolating Rickettsia telomucus from multiple sites in California. So just like want to highlight that I think when I joined PacVec in 2018, I knew nothing about vector-borne diseases. I knew nothing really about ticks. I've grown a lot within it, and it's been partly because a lot of people have been very generous with their time and knowledge. So I did want to take the time to acknowledge that I really do enjoy this community for that aspect. Oops. Let's see. Why is that zoomed in? I also want to acknowledge funders' work. A lot of PacVec, a lot of UCSB funding. So thank you. Yeah, it's fun being a tick chick, but sometimes it's not easy always finding funding. So I always appreciate anything I can find. And I think with that, thank you for listening. Here's my email if you have any questions or anything like that. And I'm still on Twitter. I know I need to merge over eventually, but that's where I'm at. So I'm happy to take some questions right now.
So yeah, thanks, Sam. Nice job. If people want to raise their hands or put questions in the chat, that would be fine. Any questions from some applause? Okay, there's one in the chat for you there, Sam.
Yep. Hi, Lars. I've been a fan of your work for many long time, so it's great getting a chat from you. So Lars asked, with regards to human encounters with nymphs, did you account for habitat type influencing risk? Yes. So part of the species distribution maps, I am bringing in a vegetation layer. So not just like the vegetation type, if it's, a woodland or grassland, but also looking at the distance from like roads and if it's in parkland versus if it's a BLM land. So I'm not quite sure if that's what you mean by habitat type, but yeah, I am trying to bring in other data layers to encompass that. I know it's not just climate that drives tick abundances and human contacts rate. There are a lot of like habitat type that's then associated with hosts and things like that.
Yeah, thanks, Sam. I was thinking mostly about the nymphs being somewhat reluctant to climb vegetation. So if you have emergent vegetation, you might get them on lizards, but they're harder to contact for people.
Correct. Yes. And so that's why the human side of this work gets a little tricky, but that's why I really have been honing in a lot of lizard work because, I know lizards are not competent hosts for a lot of these pathogens, but I think they could be a somewhat general proxy for maybe what's kind of going on in the area. Obviously, I know nymphs prefer lizards, but yeah, it's just one way to get at these kind of tricky ways of sampling these ticks in California.
The questions or comments? I'm not seeing any. Oh, there's one from Sylvie.
Hi, thanks for that talk. This is more like a general curiosity question. You were saying that, or I believe you said that if there was synchrony between the larvae and the nymphs, that there's worse disease outbreaks. Is that what you said? And I was just wondering
Yes.
why that would be then.
Yes, Sylvie, yeah, thank you for, that should have been a slide kind of talking more about the mechanism. So how it is, so for at least thinking about the Lyme disease pathogen, Borrelia burgdorferi, you have the 3 life stages of ticks, but Borrelia burgdorferi can only be acquired by infected blood meal. So if a larvae is born, it's uninfected for the most part. It doesn't have this pathogen. The only way that larvae can acquire this pathogen is through a blood meal. And so when larvae quest, they feed on a small mammal or something that may be infected with the larvae, may be infected with the pathogen. Those larvae feed, become infected nymphs. And then those infected nymphs the next year could then be infecting more and more rodents. So the idea is if larvae emerge first, way before the nymphs, they may not, larvae are not infecting new rodents. They may be picking up infection, but they're not infecting new rodents. Versus if the nymphs emerge first, they potentially could be infectious. They can be potentially amplifying the rodent community. So when larvae emerge later, they have a higher probability of taking infectious blood meal and then continuing the cycle for the following season.
Okay, so it's not necessarily the synchronicity of it, but more who comes first.
Correct. Yes. And so I think there are some pathogens that the synchrony makes more sense. So think something like anaplasma, which is another tick-borne pathogen that's hypothesized to be a short-lived infection within a host. So if you have a pathogen that's only 2 weeks in a host, you need to have that really tight overlap of nymphs and larvae to maybe amplify that pathogen. But for something like the Lyme disease pathogen, it seems like that pathogen is living in hosts longer than a couple of weeks. It could be a couple of months or their whole lifestyle. Thank you for your question.
Any other questions? I've got one that I was thinking of, which probably is because of my ignorance of sort of ticks and their behavior and how they distribute themselves. But when you have a fire incident, I imagine that the ticks that are on vegetation they're going to be toast, right? They're not going to make it out. But the ones that are attached to animals probably have a chance of escaping from the fire. Do you see any patterns in the sort of the areas around burns that indicate that
yes,
there's an increase in the because there'll be ticks that were already there, but also maybe additional ticks will start to show up and depending on what kind of host they were on, maybe they'll be further away or closer to the perimeter of the fire?
Yeah, great question. That's honestly what I got really interested in the fire project. And so if I had more time to get more nuance into this project. So, I'm thinking about 2 different time steps. I'm thinking of when the fire happens, that's probably it's probably affecting the general population right there. But I'm also thinking about the time step when the grass grows back, when the deer and the rodents and the birds come back, how long does it take for ticks to return to that particular plot? And so I think if there's a location that has lots of birds or lots of deer that can escape ticks, but then come back when the grass starts growing up, they could probably repopulate ticks at a quicker rate than, let's say, if this, it was a really, really big fire. And so it's probably less likely for those hosts to come back to then repopulate that tick population.
Okay. Thanks. Any other questions out there? All right. Well, thanks again, Sam. Nice presentation. And we'll go on to our second speaker. Yeah. Thank you.
Thanks, everyone.
Go on to our second speaker, Kacie Ring, who is also at UC Santa Barbara and also under the mentorship of Sherry Briggs and Andy McDonald. And she's getting ready to do her qualifying exams, I'm told. So good luck with those. And I remember when I did mine. So it must have been a good or a bad experience. I can't remember which it was. But anyway, good luck with your quals. And you're going to give a slightly shorter talk because I know that you've been engaged in preparing for those. And you're going to be telling us about education campaigns on tick-borne diseases. So take it away.
Yeah, thank you so much for the introduction, Neil, and fabulous talk, Sam. Good primer for talking about how to prevent or, implementing some sort of prevention method for ticks. So yeah, like Neil said, I'm in preparation for my quals, and this project is sort of out of the scope of my dissertation. So this project has not been implemented yet, but I was eager to share it with this community because it is much more on the applied social science end than what I'm normally used to. And so I'm really eager to hear people's feedback on the project design of this educational campaign. So feel free to think of criticism or notes along the way. And so this is a PacVec funded project, and it's titled Implementing and Testing the Efficacy of Educational Interventions for Tick-borne Diseases. And this is taking place in San Mateo County because I'm in collaboration with San Mateo County Mosquito and Vector Control. This is my second time being in collaboration with this vector control unit, and I really, really enjoy working with the people at this vector control agency. They just are so helpful and intelligent and have really guided like a couple of my projects. So I really appreciate their willingness to continue collaborating with me. But today, I'm just going to be introducing this project that I got funded. Once again, it is not yet implemented, so please feel free to end this talk with suggestions and critiques and questions. And so for tick prevention strategies that have been implemented, these usually occur and have been measured in high incidence states, so meaning like on the eastern coast of the US, where tick-borne diseases like Lyme disease have a higher prevalence and pose a greater risk to humans. So these are like educational campaigns like the one I'm going to be discussing today. And then they move into, testing the implementation of personal protective equipment, implementing acaricide treatments in certain regions, environmental interventions like Sam was saying, like mowing or culling of host species, and then developing vaccines and distributing vaccines to either the host community or humans. And so all of these different prevention methods vary in cost and scale, whether it be a small location to be implementing these things or statewide. But what about in low incidence states? Have these methods, interventions been implemented and then tested? And so this project is going to focus on Lyme disease and Ixodes pacificus ticks. And so in California, I'm sure most people here know that the human cases of Lyme disease is relatively low compared to the eastern and midwestern states in the US. And this is also reflective that the entomological risk of ticks being infected with pathogens is also low. And that's just due to how ginormous California is and the heterogeneity within the state. The risk of a tick having Borrelia burgdorferi can vary drastically over short geographic regions. And so I think like this low incidence but sort of awareness results in a lot of like misconceptions of actual risk of encountering a tick and acquiring Lyme disease in California. I'm sure all of us have our own personal anecdotes of things that people have told us of like people having Lyme disease, them getting a tick, their knowledge about tick-borne diseases. And this is just like throughout my years of studying ticks, it just seems like there's this wide range of knowledge of tick-borne diseases and how to prevent them and their risk of encountering Lyme disease. And so, but we all have our anecdotes, but there's no sort of actual measures of the public's knowledge of tick-borne disease and tick-borne disease prevention. And so the goal of this project is to have an intervention with the goal of changing the public's behavior. And so this is sort of designed around this theory of planned behavior, where a behavior is likely to occur if a person is confident that they can effectively perform the behavior, they have a favorable attitude towards the desired behavior, and believe that others would view them positively if they engaged in this behavior. And so with that, the goal of this project, once again, is in collaboration with San Mateo County, is to be one of the first studies in California to evaluate its residents' knowledge of ticks and tick-borne diseases in California. pilot an educational campaign that could potentially be modeled after for other counties to use in their districts. And then ultimately to improve the knowledge and attitude towards tick-borne diseases in California to make residents feel safer and actually know how to practice preventative behaviors. And so the knowledge gap is that, once again, we don't know the public's knowledge of Lyme disease and ticks in California. And so the first question that this project aims to answer is, do they have a basic understanding of tick ecology and prevention methods? And does this vary by where people live and their demographics? Does increasing their knowledge, so if they're given some sort of educational material and they learn more, does it improve their confidence in implementing protective methods and preventive practices? And does like a particular education strategy have better efficacy in improving people's knowledge and retention of information? And so my hypotheses then are that knowledge will vary by geographic location, and people that score higher in a risk assessment, so people that either live in like rural areas where they're closer to areas of high tick risk, will probably know more about ticks than people in like urban city areas. And then those residents that do have like score with a lower baseline knowledge, after consuming the educational materials, will see a greater shift in their perception and fear regarding ticks. And then participants in the study will be more likely to engage with more engaging materials such as videos and animations, leading to better materials. And I'll get more into the methods of this as we go on. And so here's the conceptual diagram of this approach for this educational campaign. And so step one is just recruiting participants, which is something else that can be evaluated. And so San Mateo County has its own sort of arm of outreach, and they have a pretty big network of people that are already of residents in the county that are already sort of in contact with them. And so they have newsletters, and then they also have individuals that can report tick contacts. And so we will go through and offer people to be a part of the study if they do report a tick contact. So if they call the county, we'll send them a tick kit and say, okay, you can also participate in the study. They have people that are signed up for newsletters, so we can recruit participants based on the newsletter. They have access to post on Nextdoor, and this could be throughout the county or it can be specific regions within the county. They also can send out mailers, recruiting people into the study, and then door hangers. And this is sort of in order of strategy. So if we don't get enough participants from the first like 3, then we'll go to like mailers and door hangers, but these are sort of last resorts. And each of these recruitment strategies will have like a QR code or some sort of identifying way to figure out how participants got recruited into the study, to see which one of these strategies works best for recruiting people into the study. And so the way we're going to implement the education campaign is sending tick kits in the mail. So we'll recruit people and then we will get their address and send them a tick kit. And within each tick kit, there will be these materials such as tick keys, tweezers, and some brochures regarding tick disease and prevention. And then within that, they'll be guided to a QR code, which will have the survey within the tick kit. And they will have their access to whatever educational material is incorporated in the study. And so originally I wanted to implement multiple types of educational materials, but I want to simplify this to reduce the amount of co-founding variables within this study. And so there's going to be 2 educational strategies. One is going to be a video that's like a tick animation video that teaches people about tick ecology and tick prevention. And the other one will be like a control, so it'll just be the contents of whatever the tick kit already has. So there is some educational materials, but it's not as obvious as like this tick animation video. And so basically people will be recruited, they will get this ticket in the mail, and then part of the ticket will have instructions for how to participate in the study. So they'll go in, they'll do the preliminary assessment, and then be guided to their educational material. Once they do the educational or review the educational materials, there'll be a post-intervention assessment, which will be the same thing as the preliminary assessment. And then following that, 1 month after the initial assessment, we'll be collecting emails through this survey, and they will be emailed a follow-up assessment 1 month later. And then the final step of this will be to evaluate the efficacy of these materials. And so that's just like an overview. And now I can discuss more about what the goal of the survey is. And so the survey is going to be broken down into a couple parts. One will be to collect demographic information on the participants and assess their baseline knowledge of ticks and tick-borne diseases, and then also assessing their behaviors and attitudes towards being able to identify a tick, how comfortable they feel removing ticks, and how comfortable they feel using a caricide and things like that. And the survey will be implemented using Qualtrics. Both San Mateo Vector Control and UCSB have subscriptions to Qualtrics, so we will implement the survey using that method. And so the survey is kind of broken down into a couple parts. First, we just need to make sure that they're eligible per the IRB approval. So they need to be above 18 and they need to live within San Mateo County. So there's just a few questions that will filter out people that don't meet those requirements. And then part 2 will be the demographics. So it'll just be, where they live, how much money they make, what education they have, kind of pretty standard demographic questions. And this is to, one, link demographics to their knowledge, and 2, see if there's any biases in people that are participating in the study. So if we're only getting people that, know a lot about ticks, that live in these rural areas that have that come into contact with ticks the most. And then part 3 will be the risk assessment. So once again, seeing where they live. So whether they live in different geographic areas that put them at high risk, what kind of job that they have. So do they have a job that puts them at a higher risk, and how much they recreate outdoors. And then part 4 will be the attitude and behaviors. So like, how comfortable do you feel about identifying a tick, removing a tick, preventing ticks? And then like, are you afraid to go outdoors because of your risk of ticks? Is something else that we'll be asking. And then part 5 will then be the assessment of like, can you identify this tick? Where do you see, like, what sort of habitat would you find a tick in? Just simple tick ecology questions. And so I'm aware of how long this survey is getting, but I'm working with San Mateo Vector Control to limit the number of questions to the most important questions so that the survey can take about 10 minutes max, because I know that people will eventually fall off of the survey if it's taking too long or if it becomes too big of an inconvenience for them. And so for the sample size for this, I ran just like a power analysis, and it seems like we're going to need at least 384 participants for the size of the county, the population size of the county. And we'll probably shoot for more because I know that people will fall off by the time we get to the like follow-up 1 month assessment. So 384 participants is the goal. And so I'll just give you guys a project status. So what is completed? Obviously created the partnership with San Mateo. I hired a scientific artist to create the educational video and wrote a script for it. And she is working on the video now. And we have designed survey questions. I sort of wrote a longer list of survey questions, and now we're just going through and parsing them down to kind of get the most bang for our buck with the length. And I've already got IRB approval for this study. And so now this project got put on hold because of my qualifying exams, but I'm actually happy that I signed up for this because it kind of put it back into my ether. And so now what we have to do is actually finish this educational materials from the scientific artist so they can be incorporated into the tick kits, move the survey into Qualtrics, and get everything set up so that it will be ready to go by the time next year rolls around. And then pilot the process. So once we get an idea of like, okay, we've got the Qualitracs survey ready to go, we have the educational materials finished, let's have some people try this survey out and see how it goes. And then that will begin the process of recruiting participants and beginning the campaign. And the goal for this project, because adult season for ticks sort of peaks in January, February, obviously depending on rain and all that kind of stuff, people will probably start beginning to get interest in ticks and tick-borne diseases in San Mateo County. So hopefully we can get this project off the ground by early February, when people start calling in and say, I found an adult tick on me, what do I do? And then we can start sending them tick kits. Okay, so that's where I'm at with this project. It's very much in the conceptual phase, beginning phase right now. But I'm really excited to get this off the ground. I think it could be really important to measure people's baseline knowledge in California. And really, like, I really want to reiterate what Sam said, like, PacVec has been really, really helpful in training me as a vector ecologist. I also was funded by PacVec in 2019 and have been able to be a part of this community, and it's great. I really, really, really have to thank the people at San Mateo County Mosquito and Vector Control. Rachel has been extremely helpful in helping me shape this project. She's incredibly knowledgeable in outreach, and I'm really grateful to have her on this project, as well as the other people at San Mateo County. Angie, Tara, Ariel, I don't have you on here, but I want to recognize you. And then Sherry, my advisor, Sam's always going to be helping me throughout this process. And then Celia and Chris, thank you for your help throughout this process. And yeah, that was a pretty rapid talk, but please ask me any questions or any comments. And if you want to talk more about this project specifically, you can totally email me and we can talk offline. But yeah, open for questions.
So thanks, Kacie. Nice job. Lars has already got his hand up, so I don't need to call for questions. Lars, go ahead.
Yeah, thank you. This is really interesting. I was just wondering if have there been any previous effort by any local agency or the state health department to produce educational videos before, or is this kind of breaking new ground? I know from the East Coast, the New York State Health Department has put out some pretty nifty videos that I think have gone on YouTube and that they're accessible from their websites.
Yeah, so there definitely there's far fewer videos on specifically like tick ecology in California, because I was trying to think if I could reuse some of the cool videos on the East Coast, but the ecology is so much different and the risk is so much different that the materials need to be suited to the West Coast. I know that Marin County has created some, Marin Vector Control has created some videos, but I'm trying to make this like, I really want to suit it towards the information that I want people to get from it. So I want more like tick ecology and tick prevention methods to be in one video. And so far I've kind of seen like parts of videos where one is about tick ecology, one is about, different prevention methods, one's about spraying a caricide in your yard. So I kind of want to like provide a video to consolidate all of that information, so people have like one thing that they can watch and understand some of the like key points in prevention.
I'm not seeing any other questions, but I think it's great that you've got a good relationship with San Mateo County, mosquito and vector control. There's a lot of those organizations in our region that's covered by PacVec, and a lot of them are engaged in really good research and are very, very helpful, I've found too. So I'm glad that you've got a partnership with them. I think it's really instructive for students to get involved and see that side of the operations side, and interact with those folks, because they're a gold mine of information.
Oh yeah, I 100% agree. My project would be nowhere if it wasn't for the help of San Mateo Vector Control. So I owe them so much for being a part of this project, and it's really, really fun to get a glimpse into the world of public health and vector control. So yeah, I'm very appreciative of this whole training process.
Okay, are there any other questions for either of our speakers today? I'm not seeing any hands raised as I scroll through, and I'm not seeing anything in the chat. So if there aren't any further questions, let's thank our two speakers again today for their excellent talks. And Kacie, good luck with your quals again. And
Thank you.
I think our next seminar is going to be after Thanksgiving on the 28th of November. And so we will see you, or hopefully many of you, back here on that day. So take care and goodbye.
Thank you, everyone.
Thanks, everyone. Enjoy your day.